University in the Department of Stem Cell and Regenerative Biology and Massachusetts General Hospital, with appointments in the Center for Regenerative Medicine, Cardiovascular Research Center, and Center for Human Genetics Research. He's an associate member of the Broad Institute and the principal faculty member of the Harvard Stem Cell Institute, where he directs the diabetes disease program and the iPS cell core facility. Professor Cowan's current focus is on using genome editing tools as therapeutics, and as a co-founder of CRISPR Therapeutics, he hopes to see these discoveries translated into treatments or cures.
Thanks for the kind introduction and for the invitation. Today I was going to talk to you about some of our efforts to use genome editing tools to try to understand more about disease mechanisms, as well as now sort of push the boundaries and maybe entering those into their own novel therapeutics. My lab actually focuses almost entirely on what I like to call the diseases that happen when you get fat, some metabolic diseases, and I'll talk to you about a couple of those today. So we're entirely focused on trying to bridge this gap from fundamental biological discovery to clinical therapy. And so my laboratory is positioned sort of in the middle here, and I'm going to try to walk you through what I think is a roadmap from getting from one end of this to the other end, and hopefully someday making a change in clinical therapies. We use the insights that come from the natural biological experiments of every day, which I like to call human genetics. And a lot of the disease genes, or genes that predispose you towards disease or protect you from diseases, are being explained every day in enormous efforts through genome-wide association studies and now through whole genome sequencing. And I'll give you an example of some of that and how we're trying to leverage those insights to then go to the next step. So even when you have the insight of, say, this gene is involved in, say, diabetes, you never know the disease mechanism, the exact way in which that gene predisposes or leads to diabetes. And that's really where my lab positions itself. It wants to know where, how, and when. And we use three powerful tools to try to get a handle on that. Stem cells, and when I say stem cells, the human pluripotent stem cells, genome editing, and the ability to turn those stem cells into any of the cell types in the human body that you might be interested in studying. If you're lucky, and you've sufficiently sort of understood your disease gene appropriately, you might, in that cell, which you created it, have a disease model, and then that model could be used to do good old-fashioned drug screens. We've done some of this drug screening, and I'm not going to talk about that today. I'm going to talk more about the genome editing and stem cells and disease modeling. And then, of course, you're never going to, if you're me, a PhD, be in the hospital delivering that therapeutic, so I really do believe that industry partnerships are critical to get all the way across. So let me walk you through our approach and some of the things we've done. So in close collaboration with one of my good colleagues, Dave Catheracin, we helped perform some genome-wide association studies to try to understand the risk factors for heart attack. And shown here is what Sage likes to call the high school, he said a high school student who designed his experiments, where essentially what he did was got patients, these are people who are genetically at risk for heart attack, and the definition for that is if you had a heart attack before the age of 40, it was almost certainly due to genetic predisposition. That's the patients. And then the non-patients are people who are much older and haven't yet had heart attacks, so they look like they might be protected. So you look across their genomes, and in genome-wide association studies, you're really looking at the single nucleotide polymorphism changes. And then you try to find if any single nucleotide polymorphism is statistically significantly found in one of the two populations. So if it's found in the patient population more significantly, it's a risk factor, and if it's found in the non-patients, it's probably protection from a particular disease. And you can plot it out like here on the bottom, which is, we're basically showing you some risk factors for heart attack. And sort of the big, tower here is 9p21, some of these others were significant. So in combination with a heart attack study, Stake also did a study of blood lipids because, as many of you know, LDL cholesterol is the leading risk factor for heart attack, and then sort of combined the two together to see what were the lipid things that were also directly associated with heart attack risk. And this was the list that we got. It felt great because it basically said the genetics works. It gives you the things that you might expect. And of course, when Joe Goldstein and Mike Brown saw this list, they were like, exactly what we've been telling you for the last 30 years. You've got everything you might want to know about LDL biology here. You've got the LDL receptor. You've got this, the target for statin. You've got the major carrier of VLDL out of the liver and protein B. So it's basically, a who's who of LDL biology. Of course, on the top here, we had one of the things that you often get from genetic studies, this interesting hit RS599A39, which really didn't mean anything. But we knew that it was important because it actually had a meaningful change in the amount of LDL cholesterol. If you were one of the lucky people who happened to be homozygous minor for this allele, you actually saw that you had a 60 milligram per deciliter lower LDL cholesterol. That's the equivalent of taking a week statin for your entire life. What that translates to is a 40% reduced risk of coronary artery disease and heart attack. So for a fairly common genetic change, that's a pretty significant change in terms of risk. Of course, most of us fall over here and are at risk. Now, we wanted to try to understand this genetic finding, and unfortunately, when we went to see where the sort of index SNP and the SNP with the highest association was, it wasn't in a gene. This is the case for 95% of these genetic discoveries from GWAS. They're not actually in genes, they're outside of genes, possibly in regulatory regions. This one actually landed in between a lot of genes in a very busy part of the genome that's in linkage disequilibrium, shown right here. And so in order to try to get a handle on what might be happening and thinking that this might be a regulatory region, we wondered, could we see whether or not some of these genes changed by genotype in tissues which were important for lipid biology? And those tissues are fat and liver. So we teamed up with Eric Schatt and also the Lee Kaplan at MGH and collected samples from the liver and subcutaneous and elemental adipose tissue. And they basically did microarrays, but also according to genotype. So shown in the red bar are people who have the homozygous major genotype, the one that is not protected, and then the blue bar is the people who would be protected from heart attack. And you can see specifically in the liver, you get changes in gene expression, a few of them. And I'm going to highlight one gene in particular because we did a bunch of work on it, sortilin, or SORT1, because it looks like it's got the direct link to LDL cholesterol. And to summarize a lot of work, what we basically found was that a single nucleotide variant created a transcription factor binding site in that person who has the minor allele. That transcription factor binding site upregulates the expression of sortilin. When you have higher levels of sortilin, you have lower levels of circulating LDL. For most of us, what it means is we have lower sortilin levels in our liver and higher LDL and a higher risk for heart attack. So here's the model that I just described, where basically it looks like this SNP is the actual causal SNP, creates a CVP transcription factor binding site, increases the level of sortilin, reduced LDL in the bloodstream. And that's through a couple of mechanisms. Turns out sortilin is a co-receptor for LDL on the surface of the liver, so it can actually take it out of circulation, but it also works just as its name implies in sorting. It sits at the trans Golgi, and it actually decides whether to take 3% for degradation, and it mostly sends them to degradation. So, we felt really good about this study. It looked like we finally made the link between a genetic insight and actual functional consequences of that genetic insight. And after, sort of patting ourselves on the back, this publication came out, which had the exact opposite finding. So when they knocked out sortilin in mice, they saw that they had reduced LDL cholesterol. This was us. We had knocked it down in the liver, and we saw higher. So we're scratching our heads wondering, what do we do from here? And I told everyone in the lab, I don't know why we're studying heart attacks or LDL cholesterol in mice. They don't get heart attacks. They have managed to do low levels of LDL cholesterol. Why don't we go back to humans? As a footnote, I will say, we figured this out. This isn't a real knockout. It was actually a hypomorphism negative, but at the time, it was very disconcerting. And at Harvard, typically when we think of studying things in human, especially at the Harvard Stem Cell Institute, we think of taking advantage of Shinya Yamanaka's Nobel Prize-winning discovery, the ability to take any human and essentially turn them into a cellular model in a dish through the reprogramming to induce pluripotent stem cells. You can then differentiate those cells into the cell type you think is affected in disease. In this case, it would be a liver cell, and then you can go from there to sort of phenotype. And in this case, our phenotype would be looking at LDL cholesterol. And it would be, the problem with this is that it's really time-consuming and expensive. And if you wanted to do this study for something that was a common genetic allele, the power calculation says that I need to do it for about 120 to 200 individuals. So you can imagine that getting the funding for that was going to be difficult. And I don't have to do it just for the people who have homozygous minor. I've got to have the controls. So it's a really large study that I have a hard time convincing people that we should do. Some other things also make it a little bit more difficult, and that's because human pluripotent stem cells and induced pluripotent stem cells, we now realize, have a lot of variation. Much of this is because of the differences in genetic background, but it's also because of differences in epigenetic state that may come from the reprogramming process. What it ultimately leads to is subtle differences between the cell lines. With the analogy I often give is if I took 10 different induced pluripotent stem cells and turned them into liver, I get 10 slightly different answers. So it means that you really have to power things to try to get the right answer. Knowing this, we sort of rethought the model. Maybe instead of doing induced pluripotent stem cell biology, we could do something that a mad scientist on the island of Dr. Moreau would do, which would get identical twins, You introduce the mutation you want to study in one of the twins, feed them the same food, have them watch the same television programs, and then see if you got a heart attack. I can't do that, but I can do almost exactly the same thing in cell culture. I can take a normal cell, make an identical twin cell that contains a mutation by genome editing, differentiate these identical twins except for the one genetic change I've made into the cell I want to study, and then do feed it to the BRCs. For me, this had a tremendous number of advantages. I didn't have to go find mutations at a hospital. I now work at the college, so my association with NGH is useful, but I didn't have to find patients and recruit them. It also meant that I didn't need to quality control the pluripotency of the cells. I knew I was working with bona fide pluripotent cell lines when I did the mutations initially. I could also do things that never really are allowed in biology. Often if you do find somebody who has a genetic mutation you want to study, if it's a pathway that you think is involved, they don't often contain multiple mutations in that same pathway, so you can try to run a static experiment. You can with this. And I will make the final argument, at least for me as an academic scientist, it's both more efficient and cheaper to try to perform these types of studies. When we first started doing genome editing, the tool that we found most useful was the transcription activator-like effectors. The ones that we used were from a plant pathogen known as Xanthomonas. It's a beautiful system where basically this plant injects these proteins, and these proteins have modular domains that allow them to very selectively bind to nucleic acid. These are pathogenic proteins, so basically they turn down the immune system of these plants. But once this sort of code was cracked by Voitis and others, we and other genome editors essentially took these and could stack them together like Lego blocks to make them bind any sequence you want. And then you could attach to them sort of any dongle or effector. And as a genome editor, the dongle you usually use is the FOC1 endonuclease. And it's made nice because it has to dimerize, so it means you get extra specificity of the DNA that you're going to bind. And once it does dimerize, it creates a double-strand break. You probably heard this morning that double-strand breaks are usually healed by non-homologous end joining, which is actually a very good process. It's not as error-prone as many people think, but occasionally it does create errors. Those indels, if they're in a gene, can be a frame shift or a knockout. You can take advantage of the other way in which cells repair double-strand breaks, which is homologous recombination or homology-directed repair, where it seeks out the sister chromatin and uses that as a copy. If you flood the cell with homologous DNA, it'll use that instead. That's the basis of homologous recombination that won Oliver Smithy's Nobel Prize. And we do that usually mostly single-stranded DNA oligonucleotides. Suffice it to say, it's a very fast process. When you deliver these pamphlets to the genome, you can basically electric break them into cells, sort them out, plate them at a clonal density, wait a few weeks, screen those colonies that you've isolated by PCR and then sequencing, and in less than a month you'll have the experiment that you want, which are these identical twins, some that are wild type and some that contain the mutation that you want to study. So, in fact, this was so easy to do, my lab got carried away. We did it way too much. I think we've done with Taylor's over 30 different mutations. We published over 17 of them in a paper a couple of years ago. And I'll get to another genome editing tool that we're using now, and I think it's exploded to hundreds, if not thousands. But I always try to remind people in the audience, just because you can edit it doesn't mean you should. You still need to use those genome editing tools to try to understand disease biology, and that's much harder. So I'm only going to be able to tell you a couple of quick stories, and in fact, just focusing in on sortilin. So we wanted to understand sortilin, that's what motivated us. And the way we did this was to, of course, edit stem cells, and we wanted to understand the role in hepatocytes, and of course, in my lab, we're also interested in adipocytes. And why was it interesting to look at adipocytes? I already told you in hepatocytes with the liver, we thought it was involved in protein processing, but there was a little report that it might actually be involved in glucose transport in adipocytes. And given that we had stem cells, we could look at multiple cell types. So we knocked out sortilin in the pluripotent cells, and this is shown here by the sequence traces. It's also proven here with a reduction in mRNA and a complete lack of protein in these knockout clones. Now we just have to turn these cells into hepatocytes. To do this, we basically rip the page out of the playbook of nature. We follow the developmental scheme that normally occurs, but reintroduce that in a dish. So this is sort of the stepwise differentiation of these cells to something that looks a lot like a fetal hepatocyte. You go through definitive endodermal, hepatic endodermal, etc., and in about 25 days you have something that looks a little bit like a fetal hepatocyte. For us, what was really nice is it has wonderful lipoprotein processing. This is just to show you what it looks like. About the end of the day, you got 85 to 95% of the cells expressing a very specific marker for the liver albumin. Okay, so now I can do my experiment, take these identical twins, turn them into hepatocytes. Now that they're identical twins, they do this with equal efficiency. There's no difference between the two. And when we looked at what the effect of sortilin was, you can see that in fact, just in line with the previous studies, you got an increase in apolipoprotein B expression or LDL secretion when you knock out sortilin without choosing control for. And And most people always tell me, oh, Chad, that's only a twofold change. Well, we know it's significant because of the 40% reduced risk, but I always try to drive home what twofold means. Imagine going home this month with half as much of your patient. That's a twofold change. So, because we're relatively careful biologists, we wanted to make sure this was entirely due to sortilin and the mutation, so we put sortilin back in the knockout cells. All three identical cells differentiate the same, and you can completely rescue this phenotype. So we're very, very convinced that sortilin is directly linked to the secretion of apolipoprotein being LDL. We then next wanted to look at fat cells, where it was thought that it played a role in these vesicles that contain the glucose transporter GLUT4 and its translocation to the membrane after insulin signal. First, we had to make fat, and I told you that most of us in the stem cell field basically follow the program of developmental biology down to sort of our final cell. The problem with this schematic is it's science fiction. There is no knowledge about the developmental biology of adipose tissue. We didn't actually know what to do to make each of these steps happen. But luckily, because of Bruce Spiegelman and his colleagues, we had a lot of information about the transcriptional cascade that was in essence essential and in many cases sufficient for making it into the site. So I wondered, maybe I could take a page out of John Manaka's playbook where you reprogram cells with transcription factors. Maybe I could program the transcription factor. So we essentially tried to put in PPR gamma into a mesenchymal progenitor cell that we could make from pluripotent cells, turn that on for about 14 days, turn it off, and ask if, 7 days later, if you've made a mature, stable hepatocyte. Luckily for us, that worked. All the work by Bruce Spiegelman over many years was true. You could very efficiently program with this transcription factor to 95% or greater fat cells in a dish. Here's what we try to do with developmental biology. Now we can look at these fat cells, and you can see this is the insulin-mediated glucose uptake in them. When you knock out sortilin, it's completely ablated. Again, we can rescue this phenotype by putting sortilin back. So not only is it important, it's absolutely required for insulin-mediated glucose transport. So what did we find out in our sortilin studies in human cells? It's absolutely required for, it's important for apolipoprotein processing, and that knockout cells have increased ApoB secretion. The function of fat cells is in fact to regulate the GLUT4 translocation to the membrane, and it's absolutely required for insulin-mediated glucose uptake. I'm going to quickly go through this one, which is a study of AKT2. It's an essential modulator of insulin signaling in important metabolic tissues such as fat and liver. In fat, when you see insulin, you increase glucose transport and lipid synthesis, and in the liver, you reduce your glucose output and increase your lipid synthesis. Why were we interested? There was an interesting publication back in 2011 of a small family that contained a mutation in this gene. These people who had the following mutation, which is a glutamate to a lysine at the 17th position, were hypoglycemic, obese, and had left side overgrowth. And why was that so interesting? It's because 10 years previous, mutations that knocked out the gene had been found, and those people had the exact opposite phenotype. The loss of function actually was hyperglycemic and decreased body fat or lipodystrophy. So it meant that maybe for the first time ever, there was a gain of function mutation in the insulin signaling pathway, and that would be really interesting to study in terms of diabetic outcomes. So we went out to study this to find out if in fact it was a gain of function mutation. Using the same system, we knocked out genes, knocked out AKT2 to do the gene knockout, and then we knocked in using a single-strand 15 oligonucleotide the exact change that was contained in the patient, which you can see sequenced here to make isolate. All right, so now we can do the experiments. Our cells that are knocked out don't have any protein. The cells that are knocked in have the equivalent amount of protein, just the amino acid change. Now we made hepatocytes. We stimulated those hepatocytes with insulin. Shown here in green is FOXO1, a transcription factor downstream of AKT. AKT normally phosphorylates that response to insulin and pulls it out of the nucleus, which you can see here by its cytosolic distribution is 15 minutes later. The knockout cells can't respond at all. And this is a true gain of function because even without any insulin at all, you can see that this E17K cell is basically pulling FOXO1 out of the nucleus very strongly. What's the consequence in liver cells? The knockouts can't turn off their glucose production when they see insulin, whereas the knock-ins basically don't produce any glucose at all, which explains the hypoglycemia that's seen in the patient. What's the consequence in fat cells? They can't really do insulin-mediated glucose uptake if you're a knockout, which is why they're lipodystrophic. Whereas these guys are off the charts in terms of how much glucose they're taking up. It's not even the same as when it's stimulated with insulin. And of course, that means that you have reduced triglycerides, which is why these people were lean, who had the knockouts, whereas the knock-in had the obesity because of the increased triglycerides. The big surprise for us, though, and what revealed something special about disease biology was that these fat cells that are constantly seeing an insulin signal act as though they're inflamed. They actually are secreting cytokines to try to recruit the immune system. And this is important in diabetes because it's been noted that this sort of inflammatory state seems to precede the onset of actual diabetes and complicate the disease. And we're very interested in finding out what it is about this signaling that's leading to an immune sort of inflamed state in the fat cells. So the fat cells look like they're the driver of this inflammatory condition. Okay, this is just a summary of what we found. Like I said, probably to me the most important thing is something that we didn't know from the patient check, which is that the adipocytes themselves seem to be involved in this inflammatory process. So for me, the real conclusion of genome editing in pluripotent cells is that you can use the technology to do a much more efficient and rigorous disease modeling. And I told you a couple of quick stories, but again, when we thought that we sort of knocked it out of the park and learned something about cells and had done something that no one had done, along comes this player on the block, the CRISPR-Cas9, and we knew about it because of our friend Zong and George Church, who had published its first use in mammalian cells. And at the time, I thought, okay, it's just another genome editing tool. And I'd used zinc fingers, meganucleases, the talents, and I thought it's just going to be, half dozen of one, 6 of the other. There wouldn't be much difference. And so one of the few times I lost the bet with a postdoc who said, no, I think it's going to be different. So I let them, I'm not going to tell you about Cas9 because I'm sure you've already heard about it. But I am going to tell you about what we did, which was try to directly compare these genome editing tools to one another. And we basically tried to set this up as the perfect boxing match, the sort of Ali Frazier. Where we had them edit the exact same alleles and the exact same cells with the exact same delivery system, everything we could to sort of match it up. And we looked across 10 different alleles. And in fact, now we've done about 15. To summarize what we found is basically this. It wasn't even a contest. It was a complete knockout. CRISPR-Cas was far more efficient. In fact, the way I usually summarize it is to say that our best TALEN was worse than our worst CRISPR-Cas. So you got efficiencies in mutation of 50 to 80%, homozygous knockouts between 7 and 25, and a full order of magnitude change in knock efficiency because of the use of Cas9. Of course, when you've got a genome editing tool that's powerful, that's sort of running around the genome, sort of cutting in a non-target way, what you immediately worry about is what's the off-target effect. Our initial look at this, which was just a gross level, what are the things that you might see by whole genome sequencing at about 40 or 50x coverage, we saw that there was nothing obvious. There were no obvious mutations that we could attribute to the Cas9, the CRISPR-Cas9 system. And we compared it with the parental as well as the family clones. The only mutation that we detected with this method was in fact a translocation in one of the tailing clones at the on-target site. So there was nothing that completely scared us. And given the efficiency, it made us start thinking this was going to transform the way we do certain aspects of biology. It's transformed the way we do disease modeling, and I'm going to tell you a quick vignette about that. It's also completely shifted the way people do animal modeling. I can tell you, I don't even request animals from my colleagues anymore. The process of shipping them, quarantining them, and getting them into your colony is far more time-consuming and costly than just making a brand new animal using the CRISPR-Cas system. And then I think it's so efficient in non-human human cells that it seems to be maybe getting to the therapeutics, and I'll try to tell you a quick story there as well. So in terms of disease modeling, remember we had this theory that this single SNP was actually the causal variant that was changing everything about fertilin expression, and then not just fertilin expression, LDL levels. And when we used to present this data, Joe Goldstein and others would be like, you haven't proven it. Those knockouts don't show me anything. You've got to actually change this SNP to absolutely prove that this is the way the genetics is working. So we set out to do that using the CRISPR-Cas system. SNP RS127403774, we've had your name. We found a guide that directly overlapped it, an RNA guide. We could eliminate just that little portion of the genome that was associated with it, and this is a non-coding region. And the result of that was exactly the result that you might expect from the EQPL. We got exactly the shift. We did it in a homozygous minor lines, and now you got a reduction in the expression of the genes, including sortilin, which directly corresponded to the sort of EQPL that we've seen previously. So we feel like we finally beat that dead horse to death. We now know sortilin and that SNP are causal. Another thing that we got inspired to do because of the efficiency of this was to try to recreate what many people call the Berlin patient. So if anyone's heard of Timothy Ray Brown, he was 2 times unlucky. He first contracted HIV and then later developed leukemia. The standard therapy for leukemia is a bone marrow transplant. His doctors knew, as many other people did, that 10 years previous to this, people had identified the co-receptor on T cells for many variants of HIV, CCR5, and that there were a very rare population of people in Northern Europe who were running around with a genetic variant from this that essentially ablated it on the surface of T cells, making them immune or resistant to HIV infection. His doctors transplanted him with bone marrow that was CCR5 deficient. That 6 years later, he's free of cancer and free of HIV. So it's sort of the human proof of concept that if you could ablate this gene in T cells and get it back into a patient or in his bone marrow cells, the hematopoietic stem cells, you could potentially cure or functionally cure HIV. We wanted to test to see if this was possible given this new technology. We did a fairly simple thing. We took mobilized peripheral blood samples, sorted out these sort of stem and progenitor cells based on just one marker, simply electroporated the system in and looked at both clones as well as batch culture what happened. Across 5 different patients, we essentially found that we could get a homozygous knockout efficiency in CCR5 of around 30%. That's just stunning. That's homozygous deleted in the cells where we delivered this. And when you talk to doctors, I talked to Bruce Walker at the Reagan Institute, he's like, this is almost clinically therapeutically ready to go at that level. Of course, when you do genome editing that's that powerful in the cell, you want to make sure that it didn't have any untoward effects. It didn't affect the way in which these cells were able to repopulate humanized mice. You could make full lymphoid and myeloid cells, and that they were definitely genome edited. Here's when we took it out of the spleen in those mice. And of course, we wanted to look in a much deeper way at the potential off-targets. Previous publications had taught us that mostly what CRISPR-Cas does wrong is in predicted ways. It's in things that look a lot like the guide RNAs. So we decided to do a very deep off-target analysis in predicted off-target sites. So we predicted, we did a target capture sequencing analysis of over 400 predicted sites. What's nice about this analysis is that you look at those sites very, very deeply, between 6,000 and 8,000 reads. So even if the mutations are 1 in 1,000, you can catch it. So first we looked at the on-target efficiency. It was even higher than we'd imagined. So we were making mutations 48% of the time in sort of our best guide conditions. So just a stunning efficiency. And when we looked hard enough, we were able to identify one off-target, one statistically significant off-target. And what that means is 36 reads that indicated a mutation out of 5,963. So a vanishingly small off-target rate that's still present. And I could have told you this off-target rate was going to occur just by looking at the guide. This one was manually designed by my graduate students, so they didn't use an algorithm that actually looks for things that have mismatches, and it's in this highly homologous gene, CCR2, that's just down the locus from CCR5 and has an exact match in its core sequence. So most people would have predicted we would have had the off-target effect that we saw. We also wanted to push this directly in vivo as a proof of concept, as a way that you might actually treat people with diseases. And the disease gene we went after is the one that's been a blockbuster for LDL cholesterol biology, thanks to Helen Hoff and Jay Schneider, PCSK9. It's actually involved in LDL receptor degradation, and if you block it, you can actually reduce the levels of LDL. And the reason we know that is that there's people out there who are knockouts for PCSK9. They have incredibly low levels of LDL and almost no risk for heart attack. So it's a blockbuster drug. We wondered, could you sort of do in vivo genome editing with this? So we delivered it into mice with Cas9 to direct PCSK9. We could basically knock it out in hepatocytes very, very well. It had a striking effect on the LDL cholesterol, lowering it, and almost no negative consequences that we could see in the liver. So in a proof of concept way, we could show that you could, in essence, get sort of a one-time vaccine and reduce LDL cholesterol for the lifetime of an animal and hopefully maybe even a human. So as this portion of the talk told us, CRISPRs are much more efficient at genome editing than TALEN. They're both very easy to assemble. We were able to show in sort of a proof of concept way that you could efficiently ablate clinically relevant genes and pluripotent human stem and progenitor cells from the blood, with a very low incidence of off-target mutagenesis, and an in vivo ablation of PCSK9 reduced plasma cholesterol, maybe providing a way to treat disease like familial hypercholesterolemia in people today. I do want to thank the people who made this possible. A lot of this work was inspired by a close colleague of mine, Karen Musa-Nuru, and a very talented postdoc, Sharon Dean. The work in the blood was done in collaboration with Derek Rossi's lab and Pankaj Mandal and Leonardo Ferrer in my lab, and then all of our off-target analysis was done in collaboration with Michael Palpas. Thanks so much for your attention. I'd be happy to answer any questions.
Two questions. One is curiosity, the asymmetry, the APT hypermorphification.
To the left, right?
Yeah.
Yeah, so the hypothesis there, I've never tested this, but the hypothesis, many of you probably know, during development, what actually establishes the left-right asymmetry is the motion of cilia that are pushing growth factors in one direction more prominently than the other. AKT is directly involved in that modal. So the idea is that it's changing functionally that, and that's why they have the asymmetric growth. I've never tested it. Somebody should test that hypothesis.
And And I guess the other one was more the technical, I guess, CCR5, compared it to the same thing during the Sangamo data.
That's right. Yeah, we compared it. So I can tell you the Sangamo data never got biallelic mutation. They had mutation rates that achieved like 33%. It would be maybe 20 to 30%, but it would be one allele that got mutated. So we're showing both alleles, complete knockout in 33% of cells. And we, in the target sequencing data, the actual mutation rate is up to nearly 50% with the guys that we use.
So then, why would
That's,
they get therapeutic?
Sorry?
Why do they get, it's only it's not biallelic.
So what they're targeting right now is there's people that are heterozygous for the CCR5 that are more common than the homozygous people. That patient population would benefit from their therapy. And we're some guys bumping around the lab, but done at clinical levels, I think you could probably do this much more efficiently than we do.
Other questions?
I was going to tease you, the speed with which you talk, I wonder if you thought about doing any commercials at the end of an advertisement.
We used to do that guy for Micro Machines. That was like,
Maybe just sort of commenting more broadly on the role of CRISPR as a therapeutic, and providing some examples that are primarily in the discovery space. You can start to see how it might be applicable in the case of HIV or maybe LDL cholesterol lowering. But maybe just sort of even a broader perspective on CRISPR therapeutics and humans, and where you see that going over the next period of years. Is it near term? Is it longer term?
So I think a lot of people, Keith spoke this morning, I didn't get to see it, but a lot of people got scared off with the initial discovery that this thing could, when unleashed in the wrong ways, could really have some devastating effects. The amount of off-target can exceed or be equal to the on-target efficacy, and that gave people a lot of pause, because you're like, okay, I'm doing a lot of unintended things as well as the intended things. Over the course of the last couple of years, we've learned how to control that a little bit better and predict how to eliminate the off-target. We can't yet predict how to get highly efficient on-target, so that's still empirically determined by most of us, but it means that it's at least as safe as the tools that are being used today, and probably even safer. So many of you may or may not know that in the gene therapy trials that are going on in blood, they're usually transplanting 10 million cells. Each one of those cells has at least one viral integration, so that means they're transplanting 10 million mutations. I think we can do better than that to cast I do think that when talking to people who do gene therapy and think about gene therapy, this is just a new widget, and we still have the same problem everybody has, which is delivery, getting to where you want to go and how you want to do it. And right now that delivery is really only solved in 3 areas: the eye, the liver, and blood. So if you want to really make a difference, think of how to deliver it to the right place at the right time in the right way. But I would guess, and probably the next speaker might agree with me, that within a couple of years you'll see some phased clinical trials using CRISPR-Cas, given its efficacy and potential safety.
Are you going to push ahead with CCR5?
I wish I could. So you would be surprised at how little interest there is in pharma for CCR5. It's because the pills, you don't want to cure HIV, you want people to take a pill. The pills are really good right now. We talked to Gilead. Gilead wasn't that interested in it. The Rayon Institute, Bruce Walker is very interested, so they are pushing ahead. They may try to go into a clinical trial, but it will probably come out of academia and not out of industry or large pharma. There's just not much appetite to treat HIV like that.
Yeah, but I guess the Sangamo path.
Yeah, that's what the regulatory path is, I would say, is definitely clear. Sangamo's already sort of shown everyone and gotten the FDA to agree on what you need to do next steps. And they may get there in a clinical trial that shows some efficacy, but I would say that this widget is the better widget. It's going to probably have more effective knockout. So I would encourage any company that could. I've been unsuccessful in encouraging the company that I'm affiliated with to do it, and we've been unsuccessful in convincing the farmers to not do it. But it seems like a no-brainer. Functionally cured from HIV.